You're welcome, my pleasure. Welcome to our session on disease, and I'm very pleased to introduce our very distinguished speakers. I begin by introducing each one of them, and then we'll have each one of them presenting for 15 minutes as we have done during the rest of the day in the two previous sessions. Welkin Johnson is a professor in the biology department at Boston College since 2011. Undergraduate at the University of California, Berkeley, earned his PhD from Tufts University School of Medicine. He was a postdoctoral fellow at Harvard Medical School and then a faculty member of the Department of Microbiology and Molecular Genetics at Harvard Medical School. With his Boston College team, Dr. Johnson studies the molecular, genetic, and evolutionary interactions between retroviruses and their hosts. Michael J. Naughton is Evelyn J. and Robert A. Ferris Professor and Chairman of the Department of Physics at Boston College. Dr. Naughton is a National Science Foundation Young Investigator Awardee and a Fellow of the American Physical Society. With an impressive list of publications, citations, and patents, Dr. Naughton's research focuses on electrical, optical, and magnetic properties of low-dimensional and nanoscale matter, including sensors, solar cells, and molecular organic superconductors. He co-founded Tau Sensors LLC and Solasta Inc. He received a B.S. in physics from St. John Fisher College, a Ph.D. from Boston University, and was a postdoctoral fellow at the University of Pennsylvania. Melissa Sutherland is an assistant professor at the Connell School of Nursing at Boston College. She practiced as a family nurse practitioner in upstate New York for 10 years, working with patients and populations with a specific focus on sexually transmitted infections and tuberculosis. Her research addresses the issue of interpersonal violence and its influence on health outcomes among adolescents and young adult women. She's a recent recipient of a grant from the National Institute of Child Health and Human Development to examine intimate partner violence and sexual violence screening in college health centers. She has been invited on the Boston Public Health Commission's Chlamydia Advisory Board, the Committee for Community Service Board at Boston Children's Hospital, and the Massachusetts Sexual Assault Nurse Examiner Multidisciplinary Advisory Board. After the presentations, as usual, we'll have time for questions. I invite you not to leave the room and stay seated because we'll have an award ceremony, and our provost, David Quigley, will host Father Leahy, the Society of Jesus, our president, for the awards ceremony. Thank you.
So, history buffs in the audience will realize right away that I've plagiarized the title of my talk, Distant Mirror, Viruses of Past, Present, and Future, from the book by Barbara Tuchman, A Distant Mirror, The Calamitous 14th Century. And Tuchman explains that title by saying that in writing about the 14th century, she felt like she was looking into a mirror that was reflecting back on our own time, so the 20th and now the 21st centuries. And that was kind of a harsh thing to say because the 14th century was dominated by more or less constant war in Europe, constant bubonic plague, and religious strife so severe that it actually rose to the very top of the church. So that was her distant mirror. The mirror that I'd like to look into goes a little bit further back in time, orders of magnitude actually. I'd like to look back millions of years and try to understand or study how viruses interact with host organisms across evolutionary timescales. And to do that, we have a problem because viruses don't leave behind fossils like other organisms, right? So what that means is if there was an interesting virus that went extinct before recorded human history, we simply wouldn't know that that virus ever existed. It would be lost to us. So that raises the issue of how do you study the natural history of the prehistoric, study viruses of prehistory. And it turns out that there are the molecular equivalent of fossils that can be generated by some kinds of viruses. And it turns out those are the viruses that we study in my lab. It's a family of viruses called the retroviruses. They're abundant in nature. Most or probably all mammalian species have endemic retroviruses. These include HIV-1 and HIV-2, the human AIDS viruses, which we also work on in the lab. Regardless of what retrovirus you're talking about, what they all have in common is that the virion of the retrovirus, when it attaches to a target cell and inserts its genetic material into the cell, they all do the same thing. They immediately change the viral RNA genome into a double-stranded DNA genome. And then they insert that DNA covalently and irreversibly into the host cell chromosome. So the viral DNA actually becomes part of the cell's DNA. Okay, so on this chromosome now you have host genes that direct the expression of host proteins, and right next to them you have viral genes that direct the expression of viral genomes and viral proteins. So these are synthesized, they assemble into new virions, and so you get the offspring of the virus then budding out of the cell, and the next generation of virus is born, and so on and so forth. The important part for this talk is this step right here where the viral genome actually becomes inserted into the host cell DNA. The other thing I need to tell you before I get to the main point of the talk is that in most organisms you can take all the cells and divide them into sort of roughly two camps or two kinds. The first kind are germline cells, and these are very specialized cells. They're the ones that become gametes, right? So they become, for example, sperm and egg. The gametes, the DNA that's contributed to the next generation, right? So the genomic DNA that's in these cells is not just the cell's DNA, it's the DNA that can be used to start the, that's passed on to the offspring of the organism. Everything else, liver cells, blood cells, muscle cells, you name it, are considered somatic cells, right? These are the tissues of the body. And usually when we talk about viruses, we're talking about retroviruses that infect somatic cells. So HIV-1, for example, infects T lymphocytes in the bloodstream and in the tissues. The DNA in these cells is the same as the DNA in these cells, except it is not inherited by the next generation. Right? So if a virus has inserted its DNA into a somatic cell, when that somatic cell dies or ceases to exist, the viral DNA ceases to exist. And that's by and large what happens during infection. But what can happen on rare occasions, extremely rare occasions probably, is that during an infection, a retroviral virion might bump up against a germline cell. And if it manages to infect that cell, then the viral DNA becomes inserted into the DNA of that germline cell. And that means that that DNA, including the viral DNA, has the potential now to be passed on as a gene to the next generation, to the offsprings of the organism, whatever organism we're talking about. Okay, so even though this is a rare event, when you consider hundreds of millions of years of evolutionary history, it turns out this has happened millions of times. And the result of that is if you look at the genomes of almost any vertebrate species, including humans, what you find is that our genomes are chock full of retrovirus-derived sequences. These endogenous retrovirus sequences are essentially molecular fossils, remnants of ancient viral epidemics that would have afflicted our ancestors hundreds of thousands or even millions of years ago. The human genome alone is estimated to have about 500,000 of these sequences. The surprising thing is that actually that number exceeds the amount of the genome that actually encodes genes and proteins. So we're more virus than we are genes. And this holds true, the level of these sequences and genomes holds true for most other species that have been sequenced. So there's a very rich fossil record. And right away, just from this information, you can conclude two things. The first is things like the current HIV-1 AIDS epidemic is not a historical anomaly, right? It actually turns out that it's business as usual from an evolutionary perspective. It's just one in a series of thousands of similar outbreaks that have occurred across millions of years. The other thing you realize when you start to ponder these kind of data, when we're talking about millions of these sequences, is you realize that viruses are not only abundant, but they have always been present. And it's almost inescapable to conclude that viruses must have had an extreme impact on the evolution of organisms, on the evolution and natural history of life on the planet, right? And I think that's an impact that is largely underappreciated at the moment. All right, so when we take these fossils, we can infer a few things by looking at them. So for example, we can guess their age based on their distribution in modern genomes. So if we look at a evolutionary tree of carnivores, for example, dogs, fox, panda, cats, and tigers, and if we find one of these viral fossils that's inserted in the same position in the genome of all 5 of these species, they share that particular fossil sequence, then that means that that virus that gave rise to that particular sequence must have infected a common ancestor way back in time, a common ancestor of all these descendant species, right? And so they all inherited that particular fossil. If it's shared by a subset of species, say just the canines, the dogs and the panda, but not found in cats and tigers, then that would be a more recent infection, something that happened in a common ancestor of canids after it split off from a common ancestor of felids, right? And the converse would also be true. The other interesting thing about retroviruses is when their DNA inserts into a chromosome, initially at the time of insertion, the ends of the viral genome have identical DNA sequences. There are about 300 or 400 base pairs of DNA on either end of the viral DNA that have identical DNA sequences, okay? And so we can take advantage of that because we know that germline DNA over thousands of generations will accumulate mutations at random with sort of more or less a clock-like regularity. So when we're looking at a modern, or I'm sorry, not a modern, but when we're looking at one of these ancient fossil sequences in a modern genome, it will have mutations such that the 2 formerly identical sequences today are no longer identical. But we can look at the amount of change, and if we have an estimate at the rate at which DNA changes, and let's say there's 15% differences between the sequences, and the rate of DNA change is about 1% per 1 million years, then we can calculate that that virus inserted its genome into an ancestral host some 15 million years ago. So we can do a molecular clock date and put ages on some of these viral sequences. All right, so what did we do? So Ted Deal, who's a postdoc in the lab, decided to look more closely at a particular set of fossil sequences, ERV-FC. So this is a virus whose sequences had already been reported by other groups to be present in the human genome. And so what Ted did is he took some of those sequences as a starting point. He started searching the genome data projects from other mammalian species, so more than 40 other mammals, looking for related or similar viral sequences in their genomes, sequences related to this particular retrovirus. For each of those genomes, each of those species where he found these viral sequences, within that species, he would take all those sequences and use them to reconstruct what the genes and proteins of the virus that infected that particular, the ancestors of that particular organism, what they looked like. Bill's waving the 5-minute sign at me. He was also able to use those sequences to reconstruct the evolutionary relationships between the viral sequences themselves, and then use some of the tricks I talked about to look at age. And so I just want to run through a little bit of his findings. So this is a mammalian family tree or phylogenetic tree. He found ERV-FC sequences in Afrotherian mammals, mammals from Africa, carnivores, a variety of different primates, lagomorphs, including rabbits and pikas, a variety of different rodents, and even one marine mammal, a dolphin. So this virus, or this lineage of viruses, was incredibly successful at jumping from one species to another and spreading among very unrelated species of mammals, actually, sometime in the past. Another noticeable pattern was looking at species that were indigenous to South America. Almost none of them had ERV-FC sequence in their genomes. In fact, the only species were primates, marmoset monkeys and squirrel monkeys. And when we look more closely at that DNA, what we can infer is the sequences that were deposited in those species were probably present in the common ancestor of those monkeys that emigrated to South America some 30 million years ago. So the virus was probably acquired in the old world and brought in sequence form to the new world with those monkeys, but the virus itself doesn't seem to have spread in South America, at least at the time. This is a tree looking at the relationships among the virus sequences themselves, and we can use this kind of analysis to identify certain patterns, and I'm just going to show you two of them. The ones in green here tell us that some species were infected more than once on different occasions by different but related strains of the same virus. So New World primates, for example, were infected at 2 different times and probably 2 different places by related viruses. So humans, a modern analogy would be in humans, the AIDS virus, there's 2 AIDS viruses, HIV-1 and HIV-2. One came from a chimpanzee reservoir, one came from a monkey reservoir, so possibly a similar situation. The other thing I want to point out is we can take trees like this and we can look for places where different species viruses are very closely related to one another. And what we're looking at here is that the viral sequences in old world primates like humans and monkeys are very closely related to sequences found in carnivores like dogs, ferrets, and pandas. And what that tells us is that sometime in the past, a virus in an ancestral carnivore was able to jump into an ancestral primate or vice versa, and probably both. And we can tell from the tree that this happened more than once. So viruses were jumping between distantly related species of mammals. So when did all this happen? It turns out enough of the sequences that Ted found, there was a significant number of them where we could look at the terminal repeats, the repeated sequences, and actually put age estimates on those. So every one of these dots on this graph is a separate endogenous fossil sequence from the genomes of the species listed on the left. And on the x-axis, we have the modern day, today at 0.0, and at the other end, 40 million years ago in the past. Okay. And what this looks like to us is that this virus appears in the genomic record and starts spreading sometime around 23 to 33 million years ago. And possibly there was a second wave of spread that began about 15 to 23 million years ago. All right, so what did the world look like 15 to 35 million years ago? One interesting point is that South America was actually still surrounded by water. It wasn't connected to any of the other continents, and that would probably explain why we didn't find viral sequences in indigenous species from that continent. This era is actually called the Oligocene epoch, and it was marked by dramatic climate change. In those days, it was cooling instead of warming. Sea levels were receding, which means that land masses that were formerly separated by water were now becoming connected by land bridges. And you can imagine what happened then is organisms, fauna that had not ever encountered one another before, were now able to migrate back and forth across continents and land masses. And so you have organisms that have never encountered each other before. Now they're encountering not only one another, but one another's viruses. So viruses now are hitchhiking along with organisms as they migrate. There was, in particular, a massive migration of mammalian species out of Asia into what we now call Europe, and at the same time, a massive extinction of the indigenous European mammals that were around at the time. The other migration I mentioned before was of a primate ancestor to South America. All right, so I mentioned in the beginning a mirror in the past looking towards the future. So what do we see there? And I can tell you, as a virologist that came of age in the late 20th century, that one of the hottest topics in virology in the last decade to 2 decades is the idea of emerging infectious disease. Right? The list of viruses that had not formally been seen before in humans, or were rare in humans, has grown incredibly just in the last couple of decades. When I was in high school, when I was about 14, I had never heard of HIV-1 or AIDS. Right? Those terms weren't even used at the time. By the time I was at a junior in high school, what happened actually that same year is my Uncle Hal became the first person I ever knew to die of AIDS. And that was 1984, the same year that the sequence of the virus was published. And within a couple of years, scientists figured out, based on that sequence and other discoveries, that the virus likely came from a chimpanzee reservoir in Africa. Somehow it jumped species and got into the human population sometime in the past. And so if you look at this list, it's a who's who of viruses that you've heard about in the news that cause disease and have come from another species in most cases. So a guanarido virus, the sin nombre or hantavirus, the Four Corners virus that you've heard about, Hendra viruses, Nipah viruses, the current Ebola virus outbreak is very likely a virus that has jumped from some other reservoir into humans. You hear about avian influenza viruses a lot now, and also swine influenza viruses. Some of you probably remember the SARS outbreak about a decade ago, and you may know that there's a related virus now called MERS coronavirus that's cropping up in humans in the Middle East, and it's thought that it's coming from camels. Right, so on and so forth. So because emerging virus infections have caught the interest of scientists, there's been a lot of examination of the distal causes of viral emergence. And some of those are predictable, but they're now confirmed. They can involve demographic change, and we heard about this earlier. Intercontinental travel, trade, and commerce means that organisms, and particularly vectors like insects and rodents, can be transported from regions of the world where they've lived for thousands or millions of years within a day, within hours or a day, to anywhere else on the planet and bring their viruses with them. And that happens and is going on even today. And you can couple that ecological and environmental changes, whether they're man-made or natural cycles. When they change the barriers that separate niches in nature, organisms and their viruses can now intermix. And that's why we're getting emerging infectious viral diseases cropping up. So this looks like history repeating itself. So I'm going to end with this slide. So this is the world today, one of those NASA photos where you can see lights all over the world. And I just want to leave with this speculative question whether or not our current era, the era of human civilization, might actually be the beginning of a new epoch that will see an increase in spillover infection, spread, and emergence of viruses. Not solely because of man-made events, but certainly involving human beings. We're now 7 billion strong, and you can see that we've spread to all corners of the planet. So I'll just end by thanking all the former and current members of the lab. In particular, I'd like to thank Ted, who's not here. He's moved on to UMass, but it was his data that I talked about. Thank you.
I'm assuming this just moves right on here. Let's see. It does. Great stuff, Welkin. I was prepared to come up and say now for something completely different, but there actually is a little overlap, not just both in science, but I use the word virus several times. Much shorter time scales. I think the goal of what I'm doing is to shorten those millions of years and those and to deal with your last slide, which is things are happening. Actually, I could put it back up. I have that ability, right? Here. Of the spillover is very rapid now. Yeah, the spillover is very rapid, and that presents new challenges to the global health community to deal with the rapid rate of spread of infectious diseases. So I've entitled this nanotech-enabled disease biomarker detection we often think of for low resource environments, but for high resource environments is also important for point of care and point of use. So we move on. I'm not going to spend a lot of time. Please don't read all these slides. We see the slides all the time of presenting the problems. We have a lot of problems. There's energy, water, food, health, security, equity, the politics end of it. And even the middle one there talks about the spread, which is leaving off from your last slide. And even the SARS, coincidentally, is in there, and the global human cost to that. My talk is about, this thing says I have 20 minutes. That's good. We're only supposed to have 15. My talk is about trying to develop tools that can detect certain things in the global health arena. So it's about public health, global health, the whole topic. Unlike our plenary speaker today, our keynote speaker, I'm not a physician, but I am a physicist. So I have to kind of explain why am I even here if I'm a physicist. I'm not, a health scientist per se. But like everybody else here, I am a doctor, and so I say I'm not a physician. I'm a real doctor, in the sense that I took Latin in high school, and I know that means teacher. So I'm going to try and tell why a physicist would even be up here. So in our lab, we do. I could define some of what we do as nanoscale integrated science for biological sensing, neurological sensing. That I'm not talking about today, clean energy, and other things. So. My job today is to say why physics and why is this integrated science and what is the virtue of nano. And I hope to do that in the next few minutes. So in a bigger picture, the old style, actually the real old style was closer to where we are now of integrated science. But the last 100 years or 150 years, the science were siloized. So there was a physics silo in the biology, chemistry, and psychology and many others, engineering even. And there wasn't none, but there wasn't enough communication between those disciplines. And I think we're realizing now, I call those the silos of silence, because there wasn't a lot of talk between them. And we're realizing now that we've always known, but there's more recognition of the virtues of integrating these scientists. And also the really important reason is we physicists know we can do a lot more with collaboration with biologists, chemists, engineers, medical technologists, etc. And so I'm going to define integrated science as more than interdisciplinary science, and I'll do my thing and you do yours, but I want your students in my lab and my students in your lab. And I think the increasing complexity of the world requires more innovative cooperation. And so it's disciplines working together to advance and even create new scientific fields, including those of today's topic that will impact global health and human health, and not just in the medical field, but in all the ones I put in the beginning of energy and water use, etc. Of course, as being an educational institution, it provides great new and exciting opportunities for our students. A quick example of integrated science would be the quantum dot, which is a phenomenon of when you make small atom-like particles, clusters of atoms that have something called quantum confinement, where the rules of quantum mechanics are governed. Chemists can use these to make things that fluoresce at different colors depending on their size, and biologists and now neuroscientists can use these to advance imaging of living systems. It's related to, it's not the same, but it's very much related to the Nobel Prize in medicine this year, which was for advances in biological imaging. And so that requires all three domains of biology, chemistry, and physics. Back to my lab, I want to make sure I acknowledge my many collaborators, some of whom are in physics, some in biology, some in psychology, postdocs, very talented postdocs, grad students, and undergrads, again in multiple departments, and a nod to the tools we make. Very much what I'm going to talk about is a certain type of tool for detecting biological diseases. Is done in the Integrated Science Nanofabrication Clean Room and the colleagues there. So I'm going to do a short, like Welkin just did. He's going to talk about one thing in his lab. I'm going to talk about one subset of things we do in our lab, and it all come from something familiar to us, the coaxial cable, which we use to get cable TV and connect to our devices in our labs. For a reason I'm not talking about today, we decided in the physics department here about 5 or 7 years ago to reduce the size of that significantly and make what we call a nanocoax. It does the same thing. It propagates electromagnetic radiation only at 10,000 times smaller wavelength, so happens to be in the visible, so we can move light on size scale smaller than the diffraction limit. And that has led to a number of publications and patents. In case there's any trustees here, they're all assigned to the trustees of Boston College. You guys own them, not me. And one subset of the use of that coax is what I'm talking about today, and that's in arrays. We make large arrays of these vertically oriented coaxes, and in addition to them being potentially really useful for nanostructured solar cells and for optical communication, they can also be useful for biological sensing and neurological sensing. But I also said I needed to say why nano. I'll try and reinforce that a couple of times, but I feel I always need, if the case is non-scientists in the audience, to set the scale for what is nano. So that's an electron microscope image of a human hair, about 1/10 of a millimeter in diameter. And so those things I just showed you there are smaller than that. In fact, if I put them to scale on that human hair, it's about that small. Or you can see it again here if I do it again. That's a closer image. That's like Tuesday, Wednesday, Thursday, Friday's hair growth. You can see the lines in them. And so we have arrays of these things that we make in the lab that are that size compared to human hair. And that's important to say because they're the size of the things we want to detect. These devices can be built on the scale of the previous talk of the size of viruses, which are about a micrometer, a millionth of a meter in size. That's about the size and spacing between these individual devices. They can actually be smaller than that. And that allows us to do many different things. If I look at the side view of a coaxial cable, again, it's just a wire inside a wire. At home, those wires are separated by Teflon. I encourage everybody to go home today, get a big scissors, go to the back of the TV and cut it. Look in, you'll see copper, Teflon copper. So we take the Teflon out, in our case it's a different material, and or we could leave it in and do different things, but if we take it out and we can trap molecules in there, because it's about the size of the molecule, so it is a significant change to any property we might measure, which is an electrical property. And we're making different things, chemical sensors, neuroelectronic interfaces. We're actually trying to develop an artificial retina where the light comes in instead of out. And the topic today is the one on the far right, is an electrochemical biosensor. But all these activities are going on in the lab with the same architecture. So if you look at that in a different viewpoint, you can imagine now having a gap between these two wires, and think of it as we're measuring electrical properties. So like the car battery, you got a plus and a minus, you got to have two wires all the time. So the two wires are the inner and the outer of the coaxial electrode. And if we can trap biomolecules, in this example is shown like antigen antibody detection, and the beauty of nature evolved in biology is that there's very specific detection of these, so very specific capture of molecules, and all the stuff that doesn't detect or that you're not looking for, that doesn't bind, can be washed away, and you're left with the things that you're looking for, the target molecules. So that's one method of looking at it. Again, talking about the clean room, this is students. Can't quite see who they are right now, but I encourage everybody to visit the clean room, go look at the website and see some of the things going on there, because all the tools we make at these nanoscale are done in our own clean room. So the one I'm going to talk about with a little bit of data is called an electrochemical sensor. This is a cartoon of an array. This is a couple of microscope images from the top down, again, looking at them. For those familiar with electrochemistry, the two battery terminals I talked about have different names. They're called the working electrode and the counter electrode. In conventional electrochemistry, these are macro scale objects. You have a metal plate somewhere, and you have another wire centimeters or longer away, and there's oxidation reduction happening at these surfaces, and ions are traveling from one wire to the other. They travel long distances over a long time and over large areas. And so we've reduced all of that to the size of a micrometer. So the center conductor is the working electrode, the outer conductor is the counter electrode, and we can make large arrays of these, but the fact that we can make arrays of these, which are have technical advantages versus the macro devices, because it's very short distances for things to move. Because they can be done at small scale, we can make arrays. We can imagine dividing this up and sensitizing each of these 12 regions to a different molecule to detect. And this whole region here, and that's about as many as you need. You know, maybe a dozen of them is enough to detect something of interest. And in that region there is, you know, 1/10 of a millimeter on a side, and that's all you would need in terms of space. So they can be extremely small. That plays later into the point of care, point of use, low resource regions where you don't need large infrastructure to do detection. You can have it, as I'll show, in principle, smartphone-based detectors. And so altogether, it's about 1 billion times smaller volume needed. Thank you, Bill, 5 minutes. I'll be done before that. We'll have more time for questions. So the key point is significantly reduced volume enables us to reduce all the electronics. The electronics world has already reduced from the cameras in your cell phone, the reduction in size of the electronics is already achieved. We want to transfer that technology for biological sensing. Don't expect you to read the labels and the axes on this, but this is a recent paper we published that shows the relative performance of this type of device versus a standard non-nanoscale planar, we call it flat surface electrode. The key point is just in the yellow here, is that we show if we scale the relative signal, it's about 100 times better, 92 times, about 100 times better performance in this nanoscale device than state-of-the-art conventional electrochemistry devices. So we're trying to parlay that now into specific biological detection. Here's an example of one is in cholera toxin. So here's comparing the state-of-the-art in biological detection. I'm not a biologist, you can correct me if I'm wrong, but one of the states of the art is enzyme-linked immunosorbent assay. This is an optical technique where you have to have significant volume of material that has been detected, and then you have illumination and microscope sources to record this. And we're doing it with all electronic detection. At the very minimum, we're getting the same sensitivity range for this particular cholera toxin. And we've taken that one step further, and we show that in versus electrochemical sensing, we can get performance that, again, this is the amplitude of the signal versus the concentration in decades. This is a logarithmic scale in both directions. But this is about 4 decades for the same signal at different concentrations, so it's about a 10,000 times improvement in performance of the nanoscale versus the macro scale electrochemical sensor. So we're obviously very encouraged, and this is a paper recently submitted with a biology, physics, biology, physics, biology collaboration. We're doing similar things where there's a third department in psychology with neuroscience developments that are next year's talk, maybe. It's an example of the value of integrated science of collaborations, because as a physicist, I don't know how to find the right molecules to bind to what molecules, but the biologists do. I know how to build the tools and how to take the measurements and to characterize these devices. So for the global health aspect, potential global health aspect, this would be an example of an ELISA instrument that you would find maybe in hospitals and in research laboratories. You probably won't find these at your primary care doctor. You won't find them in the fields in Rwanda or any other sub-Saharan Africa country because they're tens of thousands of dollars. This is like the cost of a Ferrari, maybe $100,000, including large footprint. And the device that we're developing is smartphone based. We would imagine this disposable devices. Again, we only need a fraction of a tenth of a millimeter of space to detect one target species. And you can miniaturize all the electronics, and actually to scale, that's more like about this big, you know, because that's a laptop and that's a keyboard. That's the iPhone 6 Plus. It should be even smaller. So the point here is we're able to, by using nanoscale architectures, we're able to get improved sensitivity, many orders of magnitude improved sensitivity, reduced volume of the size of our devices, and significantly reduced analyte volume, sub-nanoliters of liquid required. And so it allows for point of care, point of use in low middle income regions, and also clinical at high and other low middle income regions. So in summary, we've developed and are continuing to develop. In fact, I saw there's a poster over here by this collaboration. Good luck to Amy on the judging later. Of a nanocoax biosensor that is any number of disease biomarkers. We've chosen cholera. We're looking at many others. Proteins, we're thinking even microRNAs, of which there are some 2,000 or 3,000 microRNAs known. On a square millimeter, we could have an individual sensor for each of those once they're identified. It has prospects for pathogen detection, virus detection, improved sensitivity. The multiplex is for doing many of them. As I mentioned, very small sample volume required, and high portability, point of care, point of use. And because it's on a smartphone, it can be interfaced. You don't have to have high technical users. You can send that information to physicians at larger facilities. And with that, I want to end on time by thanking my colleagues again and for attention.
Excellent. So, I am the last speaker of the evening, officially here. So welcome. I'm delighted to be here this evening. I'm actually not going to use the word virus during my presentation, but I'm going to use another V word. I'm going to talk about violence. So I'm delighted to be here tonight to present my current research that's focused on violence screening in college health centers. As you know, I am a nurse as well as a nurse practitioner and a scientist. And I wanted to share a little bit about how I first got interested in this area, or how I came about to finding that violence against women was going to be my research trajectory. So it was about 9 years ago, and I had just started my PhD program. And I had left a clinical practice in upstate New York. I was working at a public health department where I worked in a sexually transmitted infection clinic, seeing women and men who were coming in to be tested and treated for sexually transmitted infections. And so it was in my first year of my doctoral program, and my advisor said to me, have you ever thought about the intersection of violence and sexual health? And being an honest student, I said to her no, and was sort of dismissive. I was like, I'm not sure she knows what she's talking about. But I went back to, which was a mistake, went back to my clinical practice after that first year in my doctoral program and started really looking and asking questions of the women that I was seeing in the clinic. And what I realized and I know probably most of the scientists in the room can talk about this, is kind of an aha moment. I realized how many women that I was seeing in the clinic and talking to every day and counseling had experienced violence in their lives or were experiencing violence. So I finished up that summer and went back to my doctoral program and went back to my advisor and said, you're right, this is what I want to do. I want to focus my research and my work on the intersection of violence against women, and specifically health outcomes in women. So the focus of my scholarship and research is the influence of interpersonal violence on health outcomes among adolescent and young women. And I put this figure up here because I think this really, for me, summarizes, you could replace, or you can also think about practice and policy kind of together there, but really research informs my practice informs my research. And so I really like this figure because it kind of summarizes where I'm at and where I continue to go. So a little background information. There's a little breeze up here. Intimate partner violence and sexual violence are significant public health issues, especially for female college students in the United States. Female college students ages 18 to 25 are significantly more likely to experience sexual assault compared to men, women of other age groups, and same-age women who are not on college campuses. The highest rates of intimate partner violence are also found in this age group. The health consequences of violence are numerous and significant. Sexually transmitted infections, including HIV, and major mental health issues. When we look at intimate partner violence and sexual violence, they are a national priority. 7 Healthy People 2020 objectives focus on reducing intimate partner violence and sexual violence. And most recently, the White House Task Force has put forth a white paper focused on protecting students from sexual assault. And the significance of this issue is really representing itself right now in the media, as well as other initiatives that are focusing on intimate partner violence and sexual violence. What we do know is that leading national organizations and experts recommend that healthcare providers screen for intimate partner violence and sexual violence. And that the Institute of Medicine recommends that healthcare providers screen and counsel all women for current and past IPV and sexual violence. What we also know is that in general, healthcare providers do not screen for intimate partner violence and sexual violence, even when treating women for injuries. So a woman will come to a clinic, and even though what she's presenting for is suggestive of intimate partner or sexual violence, providers are still having difficulty. There are still barriers present where they're not asking women about these experiences. And when we look at college health specifically, there's very little data out there. This study done by the American College Health Association looked at sexual violence only and relied on healthcare providers' reports, and what they found was less than 37% of college health centers include sexual violence questions in intake forms, meaning when women come into the clinic, their intake forms, their initial assessment, they're not being asked about sexual violence. And less than 49% of college health centers screened for sexual violence during GYN visits. So we have this population of women at highest risk for intimate partner violence and sexual violence. And this really led me to ask some more questions in terms of what do we know about, ask some more questions about college women. So I undertook a study about a year ago, a senior college women's study, and I looked at 615 senior college women from 2 universities in the Northeast. And I was really interested in finding out about their use of college health services and what their experiences were in college health services surrounding screening for intimate partner as well as sexual violence. And what I found was that 220 young women, about 36.1%, reported experiences of IPV and SV in their lifetime. And it matches what we see in national data. And in the past 6 months, 8.1% had experienced either intimate partner violence or sexual violence or both by a current or former partner. Now, I was also interested in their use of college health and what their experiences were in college health. 54% reported visiting the college health center at least one time in the past 6 months. And so that was, from a researcher point of view, that's good. They're going to college health. We could probably increase that, but they are utilizing. And 83.3% of participants were not asked about relationships during their most recent visit or encounter at student health. So what this told me was that over 83% were not being asked about whether they were in a relationship, what that relationship was like in terms of was it a healthy, safe relationship, and 92% reported not being asked about intimate partner or sexual violence. And again, this is despite those national guidelines that from the IOM as well as other leading national organizations. So I stepped away from this recognizing that there were missed opportunities in college health centers. Missed opportunities for intimate partner and sexual violence screening, missed opportunities to identify and refer these women, because really it's not enough just to identify someone who's experienced violence, but it's about referring and counseling and getting them into services. As I like to say, it's the warm handoff, working with patients. It's making sure they get a good referral and that it's with somebody who you can really know they're going to get good counseling and services with. Also, missed opportunities in terms of sexually transmitted infection testing and counseling. So this led me to say there needs to be a next step. And the next step was recognizing that we really need to examine healthcare providers' behaviors more. And it was identification of individual and organizational factors that influence those healthcare provider behaviors. So I wrote a grant. And it's a funded grant, so that's always a woohoo. So just last week, my grant, I heard good news. So violence screening in college health centers, that's study 2, that's the next step of my work. It is funded by the National Institute of Child Health and Human Development, and that work is underway. And it's really, the focus is to extend the work that I've previously been doing, examining women's experiences of IPV and sexual violence screening, as well as looking at healthcare providers' reports of screening behaviors, and specifically organizational factors that may be influencing healthcare providers' screening behaviors, those organizational factors that may be facilitating, as well as those that may be hindering healthcare providers' behaviors. So the next couple slides, I'm going to just talk about this study. Obviously, it's just starting up, but it will be a cross-sectional quantitative survey study. There'll be online surveys with college healthcare providers and college women from 5 colleges and universities in Massachusetts and New York. It is based upon the theory of planned behavior and specifically an organizational expansion of the theory of planned behavior. This setting and sample, all healthcare providers from 5 college health centers will be invited to take the survey. I say, I smile when I say that because I hope they all reply, but obviously they'll all be invited to participate. A random sample of 7,500 undergraduate women, 1,500 from each school, will be contacted, with the target sample being a total of 1,000 women who have been seen in college health centers in the past 3 months. Really, the goal is to get 200 women from each school to complete the survey, and that with the data from the healthcare providers as well as the organizational factors will be part of the analysis. Measures that will be looked at will include organizational factors, so healthcare providers' reports of change capacity, priority, and structure factors, as well as healthcare providers' reports of their attitudes, their beliefs, and their perceived control, as well as their intentions to screen for violence. Again, really looking at, trying to get at healthcare providers, what are some of the barriers and facilitators of healthcare providers' intention to screen. And finally, female college students will be asked details about their student health encounters. And in addition to intimate partner violence and sexual violence, whether they were asked about alcohol screening, whether they were asked about alcohol use and tobacco use. Because I really see college health centers being kind of a source or an opportunity to address these health behaviors as well as these health risks in students. So the goal is, from these two studies, is to develop future interventions to promote intimate partner violence and sexual violence screening and counseling in college health centers, and to extend the science to really understand intimate partner violence and sexual violence screening in the college health setting. And finally, to inform stakeholders. You know, a couple slides back, I think it was my first or second slide, that slide that talked about research and practice and policy. So informing the stakeholders to perhaps change or look at changing guidelines, or are there other pieces that we need to put in place to encourage providers to screen for violence. So my next couple slides, I'm going to talk about the global issue of violence against women. So violence against women and girls happens globally. Violence happens in every culture, socioeconomic group, religion, and age group. Violence against women is systemic. So when we look at globally in communities around the world, women and girls live in fear. Violence happens across the world, and this is because societies in general are shaped by patriarchal norms, which give men more power than women. And what happens then is you have issues of dependence, gender inequality, lack of education for women, and lack of power. And that leads to, or is part of the issue when we talk about violence against women. Violence against women affects women and girls at every age and every stage. And I love this. I know earlier the keynote speaker talked about the Lancet, and we didn't plan this, but I'm going to end with that. You'll see in a second here. But this is from the Lancet Web. But I really like this picture because it really just shows that violence against women, violence against women impacts girls and women at all ages and stages. And so that can include child sexual abuse, human trafficking, being sold, female genital mutilation. So this is a worldwide global problem. And so the question is, why does this matter? And this matters for numerous reasons. I'm going to outline just a couple here. Violence against women impacts the social and economic development of countries, lost productivity when women aren't able or aren't equal partners or not able to contribute to society. The annual cost of intimate partner violence, and this was in US dollars, this estimated that it's $8 trillion costs the world economy each year. It also is estimated that 1 in 3 women, this is globally, 1 in 3 women will experience physical or sexual violence at some time in their lifetime. So this is a major problem globally in communities around us. And finally, violence against women impacts the physical and social well-being of women and girls. And so this is why it matters. And this is why we need to be focusing on this issue. So I kind of tie us up here at the end with the Lancet most recently came out with a wonderful series for those that are interested in learning more about violence against women, looking at the evidence related to violence against women worldwide. And really, I'm not going to summarize the whole series, but some of the big takeaways is most of the research has been done in high-income countries. So example of my research is most of the research is being done. We need to focus more on low and middle income countries. Much of the violence research globally has focused only on sexual violence. We need to be looking at all types of violence and how that impacts not only women, but society as a totality. So to end, I just wanted to acknowledge those who have helped me along the way with my work, and to thank you for your attention. I know I was the last speaker of the day, so thank you.
Now we look forward to your questions, to take advantage of the richness of the presentations and the expertise and competence of our speakers.
This one is for Dr. Johnson. I'm wondering if this might push you out on a limb a bit. Do you expect that these ancient retrovirus segments of DNA are part of a key to unlocking some of the risk factors that we have yet to identify for modern disease, heart disease, psychological illnesses, and so on?
Well, I know there's a lot of people, because they're called virus sequences, there's almost a cottage industry of people trying to associate them with diseases. And I haven't seen anything convincing so far. So people claim that they can be associated with MS and different things like that. None of that's very convincing. It doesn't mean somebody might not find it, but I think by and large, the ones I'm talking about are so old that and degraded by mutation that they're really not functional anymore. I think that's kind of what you're asking. Yeah, so probably not a direct association. I'd be surprised.
A question here on the third row, here at the center, please.
My question will be to Dr. Johnson again. What do the factors that keep those endogenous viruses silent, or what are the factors that turn them on again? And do they share any sequence similarity between different virus types?
Yeah, so what are the factors that keep them in check? I mean,
What are the factors?
So I guess there's a couple things going on. You can imagine at the time that the sequences get into a genome, if they were at all deadly, if they produced virus at that point, the organism itself wouldn't survive. So probably most of them at the time that they're integrated, there's a strong selection against functional ones. So they may be defective right from day one. And then on top of that, it's probably the case that most organisms have evolved systems to keep those sequences silent, like methylation systems and things like that. And then I think even if they were expressed and able to transmit, probably the same sorts of genes and proteins that prevent extant viral infections might affect these as well. So innate immune systems and things like that. But by and large, the old ones are mostly defective and not an issue anymore. They haven't been pathogenic for 10 or 20 million years.
Thank you.
A question there near the end at the center. Thank you.
This question is for Mike. You did a beautiful and convincing job showing us all the benefits of these nano devices that you guys are creating, and I think it's great. Are there particular challenges that you predict that all of these types of detection devices are going to go towards the nanoscale, or do you think that there are some, like what are the main challenges involved in that, and are there some still applications in the future where we do need the larger devices?
Oh, everything's going to be nanoscale, yeah. I think there's a huge momentum in that direction. There's a lot of things besides, we're not the only ones, obviously. There's people using, I mean, what are the things you can use? You can use light, you can use electrons, you can use spin, quantum aspect. There's only so many particles we can use to detect. So we're using electrical. We're also doing some things optical. The challenges are the typical challenges of from science, from the laboratory to the device. And so there's the usual challenge of engineering things into a real product that people can use. In this particular context, it's making them, where I said we think we can go with them is truly portable, truly low cost, truly ubiquitous, disposable items and things like that. It's happening all around the world right now, and there's not, you could say there's a race on, but there's really just, everybody's just trying to get things more sensitive and more usable. And eventually companies are going to be popping up to try and market these things. We're not at that point, you know, but there's so many advantages to using the nanoscale, especially when you're making devices, tools of the size of the thing you're looking to detect.
I think we have time for one more question. Maybe a question for Melissa, Roberto, and one here, two then.
Well, Melissa, first congratulations to your R03. It's always a nice moment for a assistant professor to get those kinds of funding and the recognition for your research. With all the talk that is going on about campus sexual assault, I wonder if your study investigates potential predictors or factors that could prevent campus sexual assault, or if your study doesn't look into those, can you still talk about this as an expert? What could help us at BC to reduce the number of sexual assaults?
Yeah, thank you for the question. My study actually is focused on college health centers and looking at their role in terms of screening and then referral of women for care. But in terms of looking at college communities, I think one of as tragic as everything that's been coming out in the media is, one of the great things about it is it's really raising the level of awareness. And I think that while awareness does not equal behavior change, it is a beginning. And so we are hearing about it, seeing people are having conversations that 2 years ago we wouldn't be having. And so I think some of the conversations going on are thinking about addressing culture, campus cultures, and in order to do that, we first have to kind of understand those, and I think that's where the direction is going to be. So campus cultures, but also when we look at it as from a global perspective, it's really about changing the gender power issues, and I don't have an easy solution for that, but I do think part of that is looking at determinants of health from a broader perspective and empowering women, and there's a lot of great initiatives, and that Lancet series really focuses on that in terms of microfinancing and those sort of things in low and middle income countries. So thank you for the question.
Last question here on the right in the middle. I think it's the back.
Hi, this is for Dr. Sutherland as well. I was just wondering why college women from ages 18 to 25 are at the highest risk for sexual violence. And
thank you for the question. I think there are several factors involved in that. I think the college campus itself, their alcohol variables, being in a setting where people are living in close quarters. I think alcohol is a big predictor in that factor, but we know that they're at highest risk, and I think that's where I'm focusing right now. So certainly we can talk about that more, and I can get a better answer for you after. So.
Please join me in thanking our speakers. And I invite our provost, David Quickly, to take over and go ahead with the award ceremony.
Well, I think I can speak for everyone in attendance today. From my perspective, this has been a very truly successful event. We have seen scholarship and research from faculty across the schools and departments that, in the spirit of this university, is impacting society. Right now, we're getting to what I consider the fun part of today. In fact, in many ways, this is what I've been waiting for all day. Because what we're going to do now is we're going to highlight undergraduate student research and graduate student research that's on these posters. And what we're going to do is I'm going to introduce to you in a moment Father Leahy. And before I do, I'd like to tell you a little bit about Father Leahy. I'm sure everyone in this room knows this, but I just, for the visitors, I want to make a few points. Father Leahy is Boston College's 25th president, and he is a member of the Society of Jesus, like all the presidents before him. He arrived at Boston College in 1996, and at the time I was an assistant professor coming up for tenure to associate professor ranks, and I have to tell you, I was a little nervous. We have a new president, and I wasn't sure where I was going to fall within his radar and his range, but I think it worked out for the both of us, I might add, because since arrival, I can tell you I have worked with Father Leahy closely, probably far too many times than he would like, but particularly in the last 2 or 3 years as we have begun to think about long-term the strategic direction of this university in terms of its science and in terms of even public health. I have worked with him to discuss these issues. What I can tell you is that Father Leahy and I, and many of you, we share many things in common. He is dedicated to making this one of the great universities in this country. He is dedicated to making Boston College a leader, not just in education, which is very important, particularly to our mission, but also in what we saw here today in terms of the research and the scholarship. Since his arrival, the faculty have grown, and I believe the caliber of the faculty are the best we've ever had. I will put these faculty next to any faculty in any university in this country. More importantly, or at least equally importantly, the caliber of our students, as I was talking to a few people today, is top. It's first rate. So it is my honor to introduce Father Leahy.
If I can just stand here in front of you briefly and talk a little bit. And Tom, when I arrived and you were here, I'm from Iowa, you're from Florida, that's a great combination. You should have had no fear whatsoever. All was well. And things have gotten even better because Tom and others have taken a leading role in advancing Boston College, primarily in the area of research in the sciences. That's his focus, that's his great interest. And I would certainly reiterate his words about the enhancement of the Boston College research focus and the things that we have been able to do. When I see these posters, these boards around all of us here, it reminds me of something that the Greeks said, that philosophy begins in wonder. And I think all knowledge does. And when I look at what we're doing in the sciences and particularly trying to work on improving global health, it seems to me what we have around us and what I'm sure you heard throughout the day is the importance of certain qualities coming together. First, curiosity. Why are things as they are? Or how come things are as they are? And if you have people who have curiosity and then combine that with talent and then add on commitment, then I think you get discovery. And that's what's happening with the individuals we're going to be recognizing in a very special way today. They do have curiosity. They are talented. They are people who have commitment, and they have reached significant levels of discovery. And any great university is about doing that. Any great university is about helping the world know itself better. And in our case, given our heritage as this particular institution coming out of a Catholic and Jesuit heritage, we want that research, teaching, discovery to have an application into making the world a better place. And so when you think about the issues challenging us in global health, I think about in Africa with the Ebola virus and how that has not only affected West Africa, but all around the world. That's raised people's consciousness. So for us at BC, as we strive to be this institution that serves, that seeks excellence, we want that excellence to translate into achievement and into helping those around us. When our founder, St. Ignatius of Loyola, who founded the Jesuit order in the 16th century, moved into education, he did so because he wanted the graduates of Jesuit schools to go forth and do good, help improve the wider society. And that's what people like Dr. Childs are doing. That's what the individuals who have been engaged in these research projects as graduates and undergraduates are doing. Those kinds of people are the ones that we can look to in the future to help address all these issues that we know we have, that are challenging, no question. But I also find it encouraging that you're here and you do have that curiosity, that talent, that commitment, and you're discoverers. Some of you will be known as real pioneers. So Tom and I started here in the mid-90s. In 10 years, who knows where we will be. We've come a long way since we were founded in 1863. Much more is going to be happening because we have individuals who are really committed. So I congratulate those who have invested their time and energy into these projects, and I am looking forward to hearing more about who we are recognizing in a very particular way in the course of this evening. So Tom, as an Iowan to a Floridian, may I give this back to you? Thank you, Father Leahy.
Before we announce the winners of both the undergraduate and graduate student posters, I would like to recognize a few individuals. We have the real honor today of having both a judging panel comprised of faculty within Boston College as well as several external judges that have given their time today to be here, to go through the posters, and then to convene about an hour ago and to discuss the various posters. And I would like to recognize our judges. Rosemary Phelan is Vice Chair, Patient and Family Advisory Council at South Shore Hospital. She's a BC alum. Justine Carr is Chief Medical Officer for Stewart Healthcare Systems. Jack Connors, member of the Board of Trustees and a graduate of the Carroll School of Management. Barbara Wolf, Professor and Associate Dean for Research, Cannell School of Nursing. Amy Boski, she's a professor in the College of the Arts and Sciences, Department of English at Boston College. James Mihalkic, Professor and Associate Dean of Faculty and Academics in the Lynch School of Education. I think they deserve a round of applause for their efforts. I can tell you just a real tribute to the caliber of research that's just in this room. And it really is a snapshot of the caliber of research and the contribution of that work across the university. To give you some sense of the caliber of this work, the judges this afternoon, we had a very difficult time deciding who should receive these awards, because in the eyes of the judging panel and myself, all of the posters were first rate and deserve recognition. So what we're going to do is Father Leahy and I are going to give the awards. I will announce the winner, and we will be up front here, and some photographs will be taken, and we'll have a good time. Father? So let me begin by recognizing the award for the best undergraduate poster. The judges' comments and notes on this poster were the following. a great discussion of the healthcare implications of this work. The work was interdisciplinary in its approach. The presentation and the body of work was impressive. The title of the poster is Stimulating the Resolution of Tumor Debris in Controlled Medulloblastoma, and it is Chantel Barksdale. Where are you, Chantel? By the way, she's a biology major. Biochem, same thing. And a scholar of the college, right? Wonderful. Well, almost. It's okay. Congrats. In soliciting comments from the judges regarding the graduate student poster session, the judges for this award commented that the data were compelling, and that although current research had reached somewhat of a paradigm, that the poster and the data presented in the poster suggested new ways to move forward towards the approach to the problem. The title of the poster is Maternal Mortality, Democratization, Women's Status, and Ethnic Fractionalization, a cross-national study. And it's Mehmet Kassouni and John Williamson. And now for me, it's a real honor to announce our first recipient of Boston College's Distinguished Alumni Research Award. And I can think of no individual whose body of research is more deserving. And we saw today in his talk how important it is for individuals to make a difference in the world. And so with that, Dr. Phil Langergan. Well, I want to thank everyone for coming out. Some of you have been here all day, and that's a real tribute to your patience and dedication. And I want to thank Father Leahy for coming out today and helping us with the award. So we have a reception in the back, and please scribble in your calendars. We're going to be doing this again next year. It'll be a different theme, but I hope to see everyone here next year. Thank you very much.